Imagine a world where some of the most damaging diseases aren't caused by big predators or dramatic accidents, but by something tiny, often unseen. We're talking internal parasites. And here's a really crucial twist. Many of them need to hop between multiple different animals just to complete their bizarre kind of intricate life cycles. Welcome to the deep dive. Today, we're plunging into the fascinating and, yeah, sometimes unsettling world of what are called multisystemic parasites. Our focus is on two very distinct animal families, the Camelidae, so camels, llamas, alpacas, and the Cervidae, that's your deer, elk, and moose. And we're talking about parasites that don't just hit one organ, but sort of ripple through an animal's entire system. They lead to these widespread health issues. But here's where the story gets really compelling. Human-driven climate change. It isn't just some distant problem for polar bears. It's actually profoundly reshaping the battlefield, if you will, between these hosts and their microscopic invaders. It's creating new vulnerabilities and, frankly, unprecedented challenges. So by the end of this deep dive, our goal is for you to walk away with a really clear understanding of this intricate dance between the animals, their tiny invaders, and the shifting world around them. It's a truly crucial insight for anyone passionate about animal health and, well, our planet. Okay, let's unpack this. To really get how these parasites operate, our sources emphasize we first need to understand the animals themselves. And we're talking about two vastly different evolutionary stories here. Let's start with the Camelidae. They're true masters of survival in extreme environments. What's really remarkable about them is this strategy called adaptive heterothermy.
Right. So instead of keeping a constant body temperature like us, a dromedary camel, especially when it's short on water, can actually let its core temperature swing pretty wildly. We're talking from around 34 degrees Celsius, cool dawn, up to over 41 degrees in the scorching late afternoon heat.
Wow, that's a huge range.
It is. And the crucial takeaway here, this isn't a flaw, it's not a failure of regulation. It's a really sophisticated way to conserve water. They basically store heat during the day and then release it passively into the cooler night air. Avoids losing too much water through sweating.
Smart. And what about the South American camelids, the SACs? Llamas, alpacas.
Yeah, they also show variations, though generally less extreme. Typically maybe 35.1 degrees C up to about 39.4. Still a bit of a swing, but not like the dromedary.
Okay. This brings us to their skin and insulation, the dromedary's fur. That's pretty key too, right?
Absolutely. That thick, light-colored fur acts like a personal heat shield. It can create this remarkable 30 degrees Celsius difference between the fur surface and the skin itself. It reflects sunlight and traps a layer of air, minimizing heat getting to the body.
But for the SACs, like alpacas, that dense fleece they have for the cold Andes, that's actually a problem in the heat.
Exactly. It becomes a major liability in hot, humid climates. It just traps heat against the body, prevents the animal from cooling down effectively, and makes them really prone to dangerous overheating, to hyperthermia.
So it's a classic example of an evolutionary mismatch. What worked perfectly there causes problems elsewhere.
Precisely.
And how do they cool down differently then?
Well, dromedaries sweat right onto their skin, but beneath the fur, so the fur surface stays dry and keeps insulating. SICs, though, burdened by that thick fleece, they use specific thermal windows. These are areas on their belly, armpits, groin, areas with less fleece.
Oh, okay.
And they can expose or cover these areas just by adjusting their posture, actively managing heat release.
Clever. And of course, we have to talk about water conservation. Legendary in camels.
Oh, absolutely. They can tolerate losing up to 30% of their body water. Most other mammals, including us, would be in serious trouble, likely fatal at just 10–12% loss.
30%? How?
It's not just one trick, it's a whole multi-system biological marvel. Their kidneys have these exceptionally long structures, loops of Henle, to make incredibly concentrated urine. They reabsorb water so efficiently their feces are extremely dry. They even have special structures in their nasal passages to reclaim water vapor when they breathe out.
And the blood cells are different too, aren't they?
That's right. Uniquely elliptical red blood cells. They can handle massive shifts in hydration without rupturing, and they help maintain blood plasma volume even when the animal is quite dehydrated. It's really an incredible feat of biology.
Okay, so that's the masters of extremes. Now let's shift gears to the Cervidae deer, elk, moose. Very different picture, I gather.
Totally different. Unlike camels, they are strict homeotherms. That means they maintain a stable core body temperature within a very narrow range. They operate within what's called a thermoneutral zone, or TNZ, basically a comfort zone where they don't have to expend extra energy to stay warm or cool down.
And outside that zone?
They start burning energy, just trying to regulate.
Yeah.
And for something like a white-tailed deer in summer, Heat stress can actually begin at just 20 degrees Celsius. That's about 68 Fahrenheit.
Not very warm at all.
No, it isn't. So for them, behavior is their primary defense against heat.
Like what? What do they do?
They seek out thermal refugia. Think dense shade, like under a 90% tree canopy, or north-facing slopes which stay cooler, or areas along rivers and streams, riparian corridors. They also significantly reduce their activity. They move less, forage less, and shift these activities to cooler times, like dawn, dusk, or even nighttime.
And they use water directly, too.
Yes, standing in it, wallowing in mud if they can find it, anything to cool off.
And if behavior isn't enough, what happens physiologically?
Then their internal systems kick in. Many deer species will pant heavily to cool down through evaporation, but that increases their breathing rate and means they lose water quickly. Some larger ones, like elk, rely a bit more on sweating, and they use peripheral vasodilation, basically opening up blood vessels near the skin surface to bring warm blood closer to the air to dissipate heat.
But unlike camels, they don't have those amazing water-saving tricks.
Not at all. They are highly water-dependent. You generally won't find them more than a mile or so from a reliable water source. And their main cooling methods, like panting, are inherently water-intensive. This makes them incredibly vulnerable during droughts or dry spells.
Right, so if we connect this to the bigger picture, you can already see how these fundamental physiological differences really set the stage.
Exactly. It dictates how each animal responds not just to heat stress, but crucially to parasitic challenges that are being amplified by, well, a changing climate.
Okay, let's talk about that heat stress pathophysiology. What actually happens when they overheat?
Well, heat stress occurs when the total heat load from the environment and their own metabolism exceeds their capacity to get rid of it. And high humidity is particularly dangerous, as you said, because it really impairs evaporative cooling.
Yeah.
Sweating or panting just doesn't work as well when the air is already saturated with moisture.
Is there a threshold? Like a danger zone?
For SIs, yeah, they often use something called the temperature humidity index, or THI. A value over 150 is generally considered dangerous for them.
And what does heat stress look like in, say, an alpaca?
In SICs, you often see a rapid and severe progression, largely because of that evolutionary mismatch we discussed the fleece. Early signs might be subtle, just faster breathing, maybe over 40 breaths per minute, or being a bit lethargic, sluggish.
Okay.
But it can quickly progress to obvious distress. Open mouth breathing, drooling, clear weakness, muscle tremors, a really fast heart rate, maybe over 90 beats per minute, and a high body temperature, getting over 40 C or 104 F.
And in severe cases?
Collapse, neurological signs like seizures, confusion, and potentially death.
And you mentioned something counterintuitive about their blood.
Yeah, what's surprising is that studies have found heat-stressed camelids often show a decrease in their packed cell volume, the concentration of red blood cells. You might expect dehydration to thicken the blood, but they seem to handle fluid shifts differently. This might be linked to some of the post-mortem findings, too, like damage to skeletal muscles and even some mild nerve degeneration in the spinal cord. It's a unique response.
Okay, and what about in deer? How does heat stress manifest in them?
In cervids, it's characterized more as an energetic trade-off. Behaviorally, you see the reduced movement, less foraging, seeking shade. Physiologically, the increased panting, the elevated heart rate, the rising core temperature. But systemically, this chronic strain activates their stress axis.
The HPA axis, cortisol.
Exactly. You get elevated stress hormones like cortisol. This, combined with potentially eating less because it's too hot to forage, creates a negative energy balance. That's particularly bad during critical times like lactation, fawn growth, or antler development in males. And the ultimate consequence of this chronic stress.
A weakened immune system.
Precisely. It makes them much more vulnerable to infections, including parasitic diseases.
Okay. So we've painted a clear picture of how these incredibly resilient animals handle heat, or sometimes don't handle it so well. Now let's bring in the uninvited guest, the parasites, and see how their bizarre life cycles can wreak havoc, especially when they end up in a host that isn't equipped to fight them off. Our sources detail these specific multisystemic parasites organisms that, as you said, don't just affect one organ, but cascade across multiple systems. We're going to dive into 4 major culprits.
Yeah, and what's really fascinating here is how these parasites, despite having quite varied life cycles, often lead to this widespread systemic pathology, particularly when they encounter what we call a non-adapted host, an animal they haven't really co-evolved with.
Okay, where should we start?
Let's begin with a major neurologic nasty, the meningeal worm, Paralophostrongylis tenuis, or P. tenuis for short. This is a classic example of what we call the definitive host versus the aberrant host paradigm, meaning white-tailed deer are its natural or definitive host. In them, the parasite causes minimal, almost no detectable effect. The adult worms live pretty harmlessly in the meninges, the membranes around the brain, and the larvae are passed out in their feces.
Okay, seems fine for the deer.
Right. Then, terrestrial snails and slugs eat these larvae, becoming the intermediate hosts. Transmission happens when another grazing animal accidentally eats an infected snail or slug hidden on vegetation.
Ah, okay, so who gets sick?
Here's where it gets devastating. When animals like llamas, alpacas, moose, elk, or caribou ingest those infected gastropods, they become aberrant or dead-end hosts. In these animals, the parasite doesn't follow its normal path. The larvae migrate erratically and destructively right through the brain and spinal cord tissue.
Oh, wow.
Yeah, causing severe physical damage, mechanical trauma, and intense inflammation. This leads to a debilitating neurologic disease, cerebrospinal nematodiasis, basically worms crawling through the central nervous system.
That sounds horrific. What are the signs in, say, camelids?
In camelids, you often see focal asymmetrical neurological problems. Things like progressive weakness, especially in the hind limbs posterior paresis, general unsteadiness, ataxia, stiffness, maybe a head tilt, circling, apparent blindness, and eventually paralysis and often death.
And in moose or elk?
Similar but often profound weakness, ataxia, circling. A telltale sign sometimes is a loss of fear of humans, severe weight loss, emaciation, paralysis, and ultimately death. In fact, P. tenuis is considered a major factor in moose population declines in areas where their habitat overlaps with high-density white-tailed deer populations.
So the deer are fine, but they spread this deadly thing to moose just by sharing territory.
That's essentially, yes.
How do you even diagnose that?
It's challenging when the animal is alive. Often it's a presumptive diagnosis based on the clinical signs and knowing white-tailed deer in the area. Analyzing the cerebrospinal fluid, the CSF, can help. An increase in a type of immune cell called eosinophils, over about 17% in camelids, is highly suggestive, but a definitive diagnosis, usually only confirmed after death by finding the worms in the neural tissue.
And treatment.
Prognosis is generally guarded to poor. Prevention is key. Usually that involves physically fencing white-tailed deer out of pastures used by susceptible animals, or managing the habitat to reduce snail and slug populations.
Well, you mentioned a dilemma with treatment.
Right, the management dilemma. For valuable animals like alpacas, owners might use prophylactic deworming, say, with ivermectin every 4 to 6 weeks, specifically to prevent P. tenuis. And it is effective against P. tenuis larvae. But doing that creates immense pressure for drug resistance and other common parasites, especially gastrointestinal worms like Haemonchus.
Ah, the barber pole worm. Nasty.
Very nasty and already showing widespread resistance. So treating for one parasite inadvertently makes another, arguably more common, problem much worse. It's a real paradox of prophylaxis. What helps prevent one devastating disease fuels resistance in others.
That's a huge trade-off.
Oh.
Okay, moving on from brainworms. Let's talk about blood parasite infections. You mentioned trypanosoma.
Yes, 2 key ones here with very different stories. First, Trypanosoma evansi, which causes a disease called SURA. This one's a global threat.
Why global?
Because it's transmitted mechanically by biting flies, think horseflies, stable flies. It doesn't need a specific intermediate host like the tsetse fly, which transmits African sleeping sickness. This mechanical transmission allows it to spread almost anywhere these flies exist, and it has an exceptionally broad host range. Infects camels, horses, cattle, deer, llamas, even dogs and cats.
Who gets it worst?
Camels and horses are highly susceptible. They often develop an acute, rapidly fatal disease. Mortality can be close to 100% if it's not treated. Cattle, buffalo, and deer, on the other hand, can sometimes act as reservoir hosts, carrying the parasite, potentially spreading it, but not necessarily showing severe signs themselves.
Interesting. Has it shown up in llamas or alpacas?
You know, despite their susceptibility in theory, natural disease hasn't actually been reported in New World camelids, which is curious.
And how does SURA affect the body?
Right.
What makes it multisystemic?
It causes a whole range of problems. Intermittent high fever, loss of appetite leading to progressive weight loss, profound lethargy. Severe anemia is a big one, leading to pale gums, sometimes jaundice. You also see vasculitis, inflammation of blood vessels, which causes dependent edema, swelling in the legs, brisket under the belly. sometimes itchy skin bumps, urticarial plaques, abortions can occur, and in severe cases, neurological signs like circling, aggression, collapse, plus a general weakening of the immune system overall.
Sounds debilitating. Is it easy to diagnose and treat?
Diagnosis can be tricky because the number of parasites in the blood fluctuates, so just looking under a microscope might miss them. Serology, blood tests looking for antibodies, or PCR tests looking for the parasite's DNA are generally more sensitive. Treatment relies on a limited number of drugs, like Ceramim, but unfortunately drug resistance is becoming a growing problem for T. von C. too.
Another resistance issue. Okay, you mentioned a second trypanosome.
Yes, Trypanosoma cervi. And this one is in stark contrast to T. von C. T. cervi is actually very common in deer species worldwide, red deer, roe deer, elk, cicada, but it's overwhelmingly subclinical. It just doesn't seem to cause disease in its natural hosts.
So it's there, but doesn't really do much harm. How is it spread?
It's what's called a Stuckerarian trypanosome. Transmission isn't typically through the bite itself, but through the feces of an insect vector, like a fly. The fly might feed, defecate near the bite wound, and the host then rubs or scratches the infected feces into the skin break.
Ah, okay. A much less direct, perhaps less efficient way to get infected.
Seems that way, and certainly results in a much less pathogenic outcome for the host.
Quite a difference. Okay, let's move on to another group. The tissue cyst forming coccidia. First up, Sarcocystis species.
Right. Sarcocystis species have what's called an obligate two-host life cycle, typically involving a predator-prey relationship. Intermediate hosts, like our camelids and cervids, ingest microscopic oocysts, basically parasite eggs, from the feces of the definitive host. The definitive hosts are usually carnivores for species affecting camelids. It's often dogs or other kinemates.
So the deer or llama eats contaminated grass.
Exactly. Inside the intermediate host, the parasites undergo asexual reproduction, multiply, and then form these tiny cysts called sarcocysts, primarily in muscle tissue, skeletal muscle, heart muscle, and sometimes even in nerve cells.
And then the carnivore eats the muscle.
And gets infected, completing the cycle. The parasite then reproduces sexually in the carnivore's intestine, and new oocysts are shed in the feces.
What impact does this have on the deer or llama?
Well, most natural infections are actually asymptomatic. The cysts are often found incidentally during meat inspection if the animal is harvested. So the primary impact is often economic. Visible cysts, especially the larger macroscopic ones, can lead to condemnation of carcasses or parts of carcasses.
But can it cause actual disease?
Yes, in some cases. It's rarer, but especially in animals that are immunologically naive, maybe young or stressed, or if they ingest a very high dose of oocysts, they can develop a severe, acute, and often fatal multisystemic disease. Sometimes this is called Dalmany disease.
What happens then?
It corresponds to a phase where the parasite is multiplying rapidly and widely within the cells lining blood vessels, the endothelium, before it forms the muscle cysts. This causes systemic illness. Signs include high fever, anorexia, lethargy, myositis, that's muscle inflammation, causing tremors, stiffness, weakness, and sometimes abortion in pregnant animals. If you do a postmortem, you often find widespread hemorrhages, inflammation of the heart muscle, myocarditis, liver, hepatitis, and significant muscle damage or necrosis.
Sounds nasty. Is it treatable?
Diagnosis during that acute phase is very difficult, and unfortunately, there's no effective treatment for the chronic sarcocyst stage once the cysts are established in the muscle tissue.
Okay, and the second type of tissue cyst coccidia? Bisnoitia.
Yes, Bisnoitia species. These also have complex life cycles, often involving ungulates as intermediate hosts and carnivores as definitive hosts, although for many wildlife cycles, the definitive host isn't actually known for sure. There's also some thought that biting insects might play a role in transmission for some species.
And how does the disease look?
Bisnoitia typically causes a distinct two-phase or biphasic disease.
Okay.
Phase 1, the acute phase, sometimes called anasarca, similar in some ways to acute sarcocystis, it involves rapid parasite multiplication, particularly within the endothelial cells lining blood vessels. This leads to systemic illness with fever, vasculitis, thrombosis, clotting, and widespread edema or swelling. This acute phase can be quite severe and sometimes fatal.
And if they survive that, phase 2?
Animals that survive enter the chronic phase, sometimes called scleroderma. This is characterized by the development of very thick-walled tissue cysts, especially in the skin, the subcutaneous tissue just under the skin, and mucous membranes like the inside of the nose or eyes.
Scleroderma, like thick skin.
Exactly. It causes classic, often debilitating signs. The skin becomes thickened, folded, wrinkled, often with hair loss and the formation of small nodules. It can feel like sandpaper.
Is there a key diagnostic sign for this chronic phase?
Yes, there's a very characteristic, almost pathognomonic sign. You find numerous small, clear to whitish cysts in the scleral conjunctiva, that's the white part of the eye. Seeing those is highly suggestive of chronic besnoisiosis.
Wow. Any other long-term effects?
In males, the chronic inflammation caused by cysts forming in the testes can lead to testicular atrophy and potentially permanent sterility.
How is it diagnosed and treated?
Diagnosis in the chronic stage is usually based on those characteristic skin changes and finding the scleral cysts. It can be confirmed with skin biopsies showing the cysts, but unfortunately, like sarcocystis, there are currently no effective treatments or vaccines available for besnoisiosis.
Okay, so we've seen how these different parasites, through really diverse mechanisms, can cause widespread problems. But now, the big overlay. Climate change. Our sources really highlight how this isn't just a backdrop. Human-caused climate change is actively rewriting the rules of this host-parasite game.
Yeah, this raises a really critical question. How does global environmental change manage to simultaneously weaken the host and enhance the parasite's reach or survival? We're looking at a fundamental shift in ecological dynamics here.
So how does climate change actually shift where these parasites and hosts are found? Let's start with the meningeal worm again.
Okay, P. tenuis and white-tailed deer. Milder winters, a key feature of climate change in many areas, lead to lower winter mortality for deer. This allows their populations to increase and, crucially, expand their range northward into areas they didn't previously occupy as readily.
And since deer are the main host?
Exactly. As a definitive host expands its range, it directly expands the range of the meningeal worm. This brings the parasite into contact with populations of moose and caribou that are immunologically naive. They haven't encountered this parasite before. And as we discussed, these species are highly susceptible to fatal neurologic disease from P. tenuis. So this parasite-mediated interaction, directly facilitated by climate change enabling deer expansion, is a significant contributor to moose declines observed at the southern edge of their range.
Climate change helps the deer carry the worm kills the moose.
In simplified terms, yes, that's a major part of the dynamic.
What about the intermediate hosts? The snails and slugs?
That's affected too, but it's a bit more complex. In some northern regions, warmer, wetter growing seasons might actually enhance gastropod habitat and prolong their active season. This could increase intermediate host density and transmission risk. However, in other areas, say regions becoming more arid due to climate change, you might see a contraction of gastropod populations. This could lead to what's called an ecological mismatch, where the definitive host is present, but the necessary intermediate host isn't abundant enough for high transmission, so it's not uniform.
Right. And what about parasites spread by insects, like Sura?
Vector-borne diseases are generally highly sensitive to climate. Temperature and humidity affect insect development rates, survival, biting rates, and geographic distribution. Climate models consistently project significant shifts in the distribution of biting flies like tabanids, horseflies, and stomoxys, stable flies, under various climate change scenarios. Generally, the projection is for them to shift poleward and to higher altitudes.
Which could introduce diseases like Sura to completely new areas.
Potentially, yes, to vast new populations of susceptible hosts that have never encountered it before.
And even the ones spread by feces, like Sarcocystis and Bisnoitia.
Climate plays a role there, too. Warmer, moister conditions can enhance the survival and viability of the oocysts or sporocysts in the environment, basically prolonging the period of pasture contamination. And climate-induced changes in animal behavior can also increase risk. For example, during drought conditions, animals might concentrate around the few remaining water sources. This leads to higher densities of animals, more intense fecal contamination in those areas, and thus a higher risk of infection.
So climate isn't just moving the parasites around, it seems like it's also making the hosts more vulnerable. You touched on heat stress weakening immunity. Can you elaborate on that?
Absolutely. Heat stress acts as a potent physiological stressor. As we discussed, it triggers the HPA axis, leading to elevated levels of glucocorticoids, like cortisol. Chronically high cortisol is well known to be immunosuppressive. It impairs the function of various immune cells, dampens inflammation in some ways, but can make infections worse in others, and generally weakens both the rapid innate immune responses and the slower, more targeted adaptive responses. Basically, it makes the animal less capable of fighting off infections or keeping existing infections under control.
which leads to this idea of double jeopardy.
Exactly. That's the core concept here. You have a situation where climate change might be increasing the animal's exposure to parasites, either by expanding parasite vector ranges or concentrating animals, and at the same time, the direct effects of climate change, like heat stress, are decreasing the host's resistance by suppressing its immune system.
It's a powerful synergistic effect, hit from both sides.
Precisely. Let's go back to the moose example. Moose are physiologically maladapted to heat. They start experiencing stress at temperatures as low as 14 degrees C or 57 F. The very same warming trends that directly stress the moose are also favoring the northward expansion of white-tailed deer, which carry the lethal meningeal worm. So the moose is weakened by the climate, while its exposure to a deadly parasite increases due to climate change facilitating the deer's spread. Double jeopardy.
And similarly for, say, alpacas in hotter regions.
Yes. South American camelids, already struggling with chronic heat stress in climates they aren't adapted to, will likely have a lowered threshold for developing clinical disease from parasites they might otherwise tolerate, or parasites like P. tenuis, to which they have no co-evolved immunity anyway.
That paints a pretty concerning picture. You mentioned predictive modeling. How well can we actually forecast these future parasite landscapes? It sounds incredibly complex.
It is incredibly complex. Accurately forecasting requires modeling all the essential components of these often complicated life cycles. You need to model the definitive host, the intermediate host or hosts, any vectors like insects, and even the survival of free living stages like eggs or larvae in the environment. The parasite's potential range is ultimately constrained by the weakest link in that chain. If any one component can't survive or thrive under future conditions in a particular area, the parasite won't establish there, even if other components are suitable.
So what are the models generally projecting, despite the complexity?
Well, for P. tenuis, models project a continued potential for northward shift into Canada's boreal forest, which poses a significant threat to woodland caribou and more northern moose populations. For trypanosoma vectors, as we said, the general projection is also a shift poleward and potentially to higher altitudes. The overarching trend seems to be one of ecological reshuffling and increased instability. Climate change is essentially breaking down historical geographic and climatic barriers that kept certain hosts and parasites separated. This forces novel encounters between parasites and naive hosts, which is a recipe for the emergence of severe and often unpredictable wildlife diseases.
So given this complexity, this instability, what do we do about it? It seems overwhelming. Our sources lay out a multi-pronged approach, right? Moving beyond just treating sick animals.
That's right. It has to be about managing the host, the parasite, and the environment together. It raises the crucial question: how can we actually implement holistic, proactive strategies to address these escalating threats, both in managed populations like farms and in wild populations? It really demands integration and forward thinking.
Okay, let's start with captive and farmed populations. Llamas, alpacas, farmed deer. What's key there?
For the South American camelids especially, environmental stress mitigation, particularly heat abatement, is absolutely critical.
Like what kind of strategies?
Providing ample shade is basic, but also robust ventilation using fans, maybe directed specifically at those thermal window areas we talked about. Water-based cooling can be very effective. Misters, sprinklers, even shallow wading pools if practical. And thinking about bedding in the summer sand or cool packed earth is much better than straw, which traps heat. Similar principles apply for farmed deer. Reliable shade and access to plenty of cool, clean drinking water.
Makes sense. And parasite prevention specifically.
For P. tenuis, the most effective method remains physically excluding white-tailed deer from pastures, usually with appropriate fencing. It's not always easy or cheap, but it's the most direct way. Managing the intermediate hosts, the snails and slugs, can also help. Modifying pasture perimeters, maybe with gravel or sand barriers several feet wide, clearing thick ground cover can make those edge areas less hospitable.
What about managing the pasture itself?
Yes, good pasture management helps. Rotational grazing, where you rest pastures for long enough periods, maybe 60 to 90 days, can help break the life cycles of many parasites whose larvae die off over time. Avoiding overgrazing is important too. Keeping the forage height above, say, 3-4 inches helps because many parasite larvae are concentrated in the lowest couple inches of the grass near the soil.
And co-grazing, I've heard of that.
Co-grazing with a non-host species can be beneficial. For example, grazing horses or cattle alongside camelids. The horse or cow might ingest parasite larvae that are specific to camelids, but since they aren't suitable hosts, the parasite life cycle is broken. They act like biological vacuum cleaners in a sense.
Interesting. What about just basic animal health?
Crucial. For llamas and alpacas, annual shearing before the hot season starts is vital for heat stress prevention. Ensuring balanced nutrition with adequate protein and essential trace minerals helps maintain a robust immune system, and biosecurity is key. A strict quarantine protocol, minimum 21, ideally 30 days for all new animals coming onto a farm, is essential. During quarantine, they should be tested for relevant diseases and parasites and treated appropriately before mixing with the resident herd. This prevents introducing new problems, or perhaps even worse, introducing drug-resistant strains of parasites.
Right. Which brings us back to the drugs and that paradox of prophylaxis. How do managers navigate that?
It's tough. As we said, routinely deworming camelids every 4-6 weeks with something like ivermectin is effective at preventing P. tenuis. But the cost is enormous selection pressure driving resistance in gastrointestinal nematodes like Haemococcus contortus. This really underscores the urgent need for effective non-pharmacological prevention strategies for P. tenuis, like the fencing and habitat management.
So what's the approach for the gutworms then, if not routine deworming?
For gastrointestinal nematodes, the current standard of care, the best practice, is moving towards targeted selective treatment, or TST. This means you don't treat every animal. Instead, you identify and treat only those individuals who are clinically affected, e.g., anemic, losing weight, or who have demonstrably high parasite loads, often determined by doing fecal egg counts or using tools like the Famicase system to assess anemia levels by looking at mucous membrane color.
Why only treat some?
The key idea is to preserve refugia. That means maintaining a portion of the parasite population on the pasture that remains susceptible to the dewormers because they haven't been exposed. These susceptible worms reproduce and dilute out any resistant genes that arise in the parasites within the treated animals. It slows down the development of resistance significantly compared to treating everyone.
That's a huge shift from the old way of just blanket treating everything.
It is, but it's essential for preserving the effectiveness of the few deworming drugs we still have left.
Okay, that covers managed populations. What about wild populations? Direct intervention is much harder there.
Much harder, often impossible on an individual level, so the focus shifts to habitat and population management. Protecting and restoring those thermal refugia we talked about, dense forests, areas along rivers, north-facing slopes, becomes critical for allowing animals to cope behaviorally with increasing heat.
And managing animal numbers.
In some specific situations, managing host densities might be considered. For example, where excessively high white-tailed deer populations are clearly implicated in the decline of a vulnerable native species like moose due to P. tenuis transmission, wildlife agencies might consider population control measures for deer to reduce that transmission pressure. It's often controversial, but sometimes deemed necessary.
What about the interface between wildlife and livestock?
That's a critical zone for surveillance. Wild deer, for instance, can act as reservoirs for drug-resistant parasites that can then spill over into farmed deer or small ruminants sharing the same areas. And domestic dogs, if allowed to roam and scavenge carcasses of wildlife, can potentially pick up Sarcocystis or Besnoitia and then shed oocysts back into the environment, perpetuating those cycles and potentially exposing livestock.
And moving animals around. Translocation.
Translocation policies need to be really careful. Any plan to move animals, like translocating white-tailed deer for population establishment or hunting purposes, must include rigorous parasite screening and a thorough risk assessment. We absolutely want to avoid situations like accidentally introducing P. tenuis into Western North America, where native cervids like mule deer and elk have no immunity.
So looking ahead, wrapping this up, what are the top priorities for research and surveillance to get a better handle on these complex, shifting challenges?
Well, there's definitely a high priority need for improved diagnostics, particularly sensitive and specific tests that can be used on live animals, antemortem tests for P. tenuis and those aberrant hosts like moose and alpacas, and also better diagnostics for the acute phases of sarcocystosis and bisnoisiosis. We also need more research to really quantify how environmental stressors, heat, nutritional stress interact to modulate immune responses and actual susceptibility to these different parasites in different species.
And the predictive models.
Refining those predictive models is crucial. They need to incorporate more detailed ecological data on the specific intermediate hosts and vectors involved, their climatic tolerances, and how land use changes interact with climate change. And finally, we really need to move towards more integrated surveillance systems, not just passively reporting diseases when they pop up, but actively integrating monitoring of host population health and distribution, parasite prevalence, and spread, perhaps using modern molecular tools on environmental samples and tracking relevant climatic variables. This could help build effective early warning systems for emerging threats.
Wow. We've definitely covered a lot today. We've journeyed through the unique physiology of camelids and cervids, these masters of adaptation in their own ways. We've unpacked the complex lives of several damaging multisystemic parasites, the meningeal worm, trypanosome, Sarcocystis, bisnoitia. And critically, we've seen how climate change is acting as this profound threat multiplier, creating novel, unstable, and frankly worrying disease landscapes by messing with hosts, parasites, and their environments simultaneously. As we consider these intricate interactions between large animal physiology, these microscopic parasites, and huge global climate trends, it really makes you wonder, doesn't it? What fundamental responsibility do we as a species have in trying to maintain the delicate balance of these evolving ecosystems, especially given our undeniable role in accelerating so many of these changes? Something to think about. Thanks for joining us on this deep dive.